Hi there, I am Benit Maru. I am an adult neurologist and currently the Chief Medical Officer and Head of Clinical Development at Taysha Gene Therapy. Before proceeding further into this talk, I need to make sure everyone is aware that what we are discussing here is an investigational gene therapy product. Until clinical trials are completed and regulatory agencies such as the European Medicines Agency and the US FDA potentially approve a product, we cannot make any claims about the potential safety and efficacy of an investigational product. Our goal here is to educate about our investigational clinical trial. Taysha is focused on the development of gene therapies for central nervous system disorders. The strategy is building upon one of the most well-studied delivery vehicles, the AAV9 vector, adeno-associated virus. The therapies Taysha are developing target the underlying biology of a very specific condition, and Taysha utilizes an intrathecal delivery, which is an injection into the lower part of the back into the cerebrospinal fluid. At Taysha, we work with advocacy groups and seek input on materials and trial protocols. We wanted to understand the journey of diagnosis to Rett syndrome. What are some of the most challenging symptoms? What's the significant burden and unmet needs? We also wanted to understand the variation in Rett syndrome experience over time. We also wanted to understand and gather input from the community regarding just clinical trials in general and overall expectations. Now, this work here was a two-pronged approach. There was a quantitative survey, multiple choice questionnaires, participants from 4 countries. The second piece was slightly more in-depth. There was a daily survey, there was a guided discussion, there was a focus group as well, and it was driven by 20 caregivers in the US. Now, what you can see here is a brief overview of some of the impacts that we garnered from the work within the community. There's lots of symptoms here which do cause impact, loss of speech being one of them. This was specifically centered around caring for an adult with Rett, and this has obviously led to us implementing certain endpoints within our current adult trial as well. So now moving on to gene therapy. What is it and how could it work? Every gene in the body makes a specific protein, and these proteins ensure our body works appropriately and properly. Unfortunately, there are lots of diseases that can occur when genes are impacted. Here we are specifically going to be speaking about monogenic, so one gene causing diseases. There are a multitude of ways that a change in a gene can actually cause a problem. For example, a gene change can result in a non-functioning or even missing protein, and this occurs in diseases such as in certain neuromuscular conditions. There is also the potential when a gene can go wrong that it can produce too much protein, which can that in itself then be harmful. For example, there are genetic eye diseases which cause blindness due to too much protein, which becomes toxic, and unfortunately people lose their sight. And also fibrosis of the lung as another example where there is too much protein. Now, some gene therapies work by delivering new copies of the gene or by helping to silence the protein that is being overproduced. These genes are delivered to the cell inside the body by various delivery routes, with Taysha's primary choice route of administration being the intrathecal mode of delivery. So a quick overview on Rett, as I'm sure you're all aware. MECP2 is the gene that tells cells how much MECP2 protein should be made. Now in Rett, a reduction of MECP2 reduces normal protein in the brain and nerve cells. Unfortunately, on the other side of it, there is another condition, which is MECP2 duplication syndrome, where when you have too much MECP2, this can cause signs and symptoms of a disease as well. And this is known as the Goldilocks phenomenon, where too much is harmful and too little causes Rett syndrome. So this is that balance between too much protein being expressed and not enough protein being expressed, as talked about in the slide before, when something happens or something goes wrong with a gene. So Taysha's gene therapy delivers a working copy of the MECP2 gene to the affected areas. And now we know that there is a concern of too much MECP2, which could cause a duplication syndrome, and it can cause challenges in itself. Now, our extremely talented collaborators developed technology, which I personally think is quite cool, which can help control the expression of protein and minimize the harm from overexpression, but also by helping control the cells which do not have enough. So it can help increase MECP2 protein in the cells that do not have enough, and it can stop cells from producing too much, where it could go wrong, potentially. The gene is put in a delivery vehicle. The delivery vehicle of choice for Taysha is the AAV9. Now, the AAV9 as a packaging, think of it as an envelope, has a certain size. Due to that size restriction, and because of the additional control that we're putting in with the miRare technology, which helps us control for any overexpression, that with the MECP2 gene, it's not all going to fit within the AAV9 envelope. Because of that, certain parts of the MECP2 gene were removed, and the most important parts to give you a functional working MECP2 protein were kept, which allows us all to fit into this one envelope. Now, based on all of our non-clinical data packages, there have been no adverse events that have been seen, and overall, we can surmise that the product does change outcomes in the mouse model. The therapy will be delivered via an injection in the lower back, and once the product has been injected into the cerebrospinal fluid, it will travel up the spinal cord and it will be transferred to the cells ultimately that need it. So we do currently have an adult study ongoing. It is an open label phase 1-2 dose escalation study, primarily a safety and tolerability study with an early efficacy signals to be monitored during over 2 dose levels. This will be the first time any person with Rett will have been treated with a gene therapy. And our first site is located in Canada. What are our plans for the broader Rett syndrome community? So firstly, as we mentioned, we have an active site in Canada. The dosing of the first adult patient is expected to be the first half of this year, 2023. Initial data from the study is expected to be shared from the first half of 23, and quarterly updates on clinical data, which will primarily be safety, will be given thereafter. With regard to our pediatric plan, we plan to go to the United Kingdom, we plan to go to the MHRA and submit a clinical trial application by the middle of this year to allow us to start a study in pediatric females with Rett syndrome. With regard to the US, we plan to submit an IND in the second half of this year. And for males with Rett, currently at this time, Taysha is primarily focused on running preliminary clinical trials in adult and pediatric females with the hope to demonstrate potential safety and efficacy of this investigational product. We do have plans for global clinical trials, and if there are any questions, please do email medinfo@tayshagtx.com. Now, finally, a great big thank you. Thank you to Rett Syndrome Europe and to all other advocacy groups that have supported us, worked with us, partnered with us. Massive thanks to Sarah Sinnott, Stephen Gray, and Berge Manassian at UT Southwestern. These are the extremely smart collaborators who I think have created a rather nuanced, novel, and intriguing piece of technology. And thank you to the entire community to help partner with us, work with us, and help us bring this therapy forward. Thank you.